Welcome to this lecture on insect vectors and diseases. Today we are going to explore a fascinating and crucial concept in medical entomology known as insect reservoirs. If you have ever wondered why some diseases seem impossible to wipe out, even with modern medicine, you are in the right place. Let us dive in and uncover the hidden survival strategies of these pathogens. To start, imagine you are trying to empty a flooded room using just a bucket. You work hard, but there is a hidden pipe constantly pumping water back in from beneath the floorboards. No matter how much water you remove from the surface, the room will never stay dry as long as that hidden source exists. In public health, we face a very similar problem. We can treat every single sick human with medicine, but the disease might mysteriously return the next year. This happens because the pathogen is hiding in a secret storage tank in nature, which scientists call a reservoir. Now that we understand the idea of a hidden storage tank, let us define what this means in medical entomology. An insect reservoir is an insect population that sustains a disease-causing pathogen over an extended period. The most crucial part of this definition is that it happens completely independent of a vertebrate host, like a human or animal. The insect becomes a long-term biological host, where the pathogen lives, multiplies, and survives across different seasons and generations, even when there are no sick humans around to bite. Building on that definition, it is important to distinguish between a simple vector and a true reservoir. Normally, we think of insects merely as vectors. A vector is like a flying syringe that simply carries a disease from one infected person to a healthy person. It is a temporary transporter. However, when an insect acts as a reservoir, it serves as a permanent biological home for the pathogen. This means the disease can survive perfectly well even if all vertebrate hosts in the area are immune, vaccinated, or completely absent. This brings us to an important question. How exactly does the insect maintain this disease across generations without biting an infected human? The answer lies in a biological process called vertical transmission, specifically known as transovarial transmission. In this process, an infected female insect passes the pathogen directly into her eggs. Consequently, the newly hatched offspring are already infected from birth. They do not need to bite a sick person to acquire the disease. They are born capable of spreading it. This ensures the pathogen's survival generation after generation. Understanding this vertical transmission mechanism is absolutely critical for global disease eradication programs. If a health organization only focuses on curing humans, they will ultimately fail to stop a disease that utilizes an insect reservoir. For example, dangerous viruses like dengue and Zika can be maintained in mosquito populations during dry seasons or harsh winters. The adult mosquitoes may die off, but their infected eggs survive dormant in the soil. If we did not know about this reservoir mechanism, we would be completely baffled when the disease suddenly reappears after the rains return. Therefore, identifying these reservoirs forces scientists to design comprehensive strategies that target breeding grounds. To truly understand how a disease survives inside an insect reservoir over multiple generations, we can use a mathematical model of population dynamics. This formula calculates the number of infected insects in the next generation, denoted as IN+1. It depends on 4 factors. First, IN stands for the number of infected adult females currently present. F represents the average number of eggs laid by a single female. S is the survival rate of those eggs to adulthood, expressed as a decimal. Finally, V represents the vertical transmission efficiency, which is the percentage of eggs that successfully inherit the pathogen from the mother. If either the survival rate or the transmission efficiency decreases, the number of infected offspring will drop significantly. Now that we have our formula, let us see this with a concrete example. Imagine a population of Aedes mosquitoes acting as a reservoir for dengue. We are given 100 infected females, each laying 50 eggs. The survival rate is 20% and vertical transmission is 10%. We substitute these values into our equation. First, multiplying the females by their eggs gives 5,000 total eggs. Next, multiplying the survival and transmission rates gives a combined success factor of 0.02. Finally, multiplying the eggs by this factor yields exactly 100 infected adults in the next generation. This means the disease is perfectly maintaining itself. This stable baseline leads us to an important question. What happens if we intervene? Let us look at sandflies carrying leishmaniasis. We start with the same 100 females, 50 eggs, and 10% transmission. However, health workers spray insect growth regulators, dropping the survival rate to just 5%. We plug these new values into our formula. The total eggs remain at 5,000. But our new combined success factor is much lower, at 0.005. Multiplying the 5,000 eggs by this tiny fraction gives us a final answer of just 25 infected sandflies emerging in the next generation. So we have calculated the new population size. This brings us to the mathematical proof of why targeting breeding sites works. We started with an initial population of 100 infected insects. After the intervention, the next generation only produced 25 infected adults. By comparing these two generations, we see that 25 is strictly less than 100. Mathematically, this means IN plus 1 is less than IN, indicating that the population of infected insects is shrinking rather than growing or staying stable. Continuing from our comparison, we can now state our final conclusion. Because the next generation is smaller than the current one, the reservoir is no longer stable. It is actively collapsing. This proves physically and mathematically that reducing larval survival is a highly effective way to flush the pathogen out of the insect reservoir. Over a few generations, this continuous collapse will eventually lead to the local eradication of the disease, proving that environmental control is far more effective than just treating human patients. Now that we have proven how to collapse a reservoir, let us visualize the entire process. In this diagram, you are looking at the insect reservoir maintenance cycle. Notice the circular path connecting the adult female, eggs, larvae, and pupae. The most critical part of this diagram is the arrow labeled vertical transmission, pointing directly from the adult female to the eggs. This shows the pathogen being passed on before birth. Over on the right, you see a faded human figure bypassed by the main cycle. This perfectly illustrates our core concept. The disease survives and loops continuously within the insect population, completely independent of the vertebrate host. We have covered a lot of ground today, so let us review the key takeaways. First, insect reservoirs act as biological safe havens, allowing pathogens to survive completely independently of vertebrate hosts. Second, this survival is made possible through vertical transmission, where insects pass the infection directly to their offspring. This explains why diseases can persist through harsh seasons and aggressive medical treatments of humans. Finally, understanding this shifts our entire public health perspective. To permanently eliminate the hidden source of a disease, we must actively manage and interrupt the reproductive cycles of the insects themselves. Great job following along with these concepts today.